Alrighty. So this talk has been updated since the last time I gave it. I've got some new information. I think you'll find this interesting here. Amber and honey. So most people think of amber arising from the sap in the tree, but that's not correct. Here you see the vessels which allow for fluids to go up and down in plants, especially trees here. And the xylem is what contains the watery fluid with water and, say, dissolved minerals such as potassium or sodium. And the phloem are the channels through which the sugar is transported up and down. But those make up what we call sap and are different than resin. Resin is actually a special substance composed of many different molecules that are specifically for healing the wounds to the tree, by whatever mechanical means or an animal chewing on it or something. So it's a very different substance. And it's seen through an analysis of the various specimens from around the world that the majority of amber has substance from trees, especially such as what's called the kauri tree, which is a very unusual type of pine, which is still growing today in that northern region of New Zealand. And here's the shape of the cone. So it's actually more like a globe-shaped cone than the more typical ones that we see. And it has these very unusual arrangements for the leaves like this, as opposed to the usual coniferous trees that we see with the more typical shaped pine cone. But also these trees, these extant trees, the ones that we see today, are also ones that have contributed the resins for the amber specimens that are found in various parts of the world as well. So this means that a significant amount of amber comes from deciduous trees with flowers, which means flowering plants. And this is a major problem because the first flowering plants, supposedly according to the evolutionary time frame, arrived around 130 million years ago. However, amber formation is thought to have occurred anywhere from some 23 or 20 some to 320 some million years ago. So there's this problem of this 100 years of amber from trees with flowers before flowers are supposed to have evolved upon the scene. So this is a significant problem for the evolutionists' time frame. Well, this is the worldwide distribution of the major locations of amber as identified. And so I'll show you, highlight a few of them, such as from the Dominican Republic here, the northern shore of the island of Hispaniola. And so it's especially remarkable for the various unusual colors that are seen in addition to these usual colors you see on the screen now. You also get these ambers with blue and green, which is quite remarkably different than the more common specimens. In Mexico, from the state of Chiapas in the very south there are these multicolored specimens that are found. And then up in Canada are these blood red specimens that originated in the lower corner there, the southeastern corner of Alberta, but were transported by some mechanism. We know what that is, but the evolutionists have a difficulty with it, over to the place that's further to the northeast. Burma is also a location for many remarkable specimens which entrap species of animals, especially that are not found elsewhere in the world. And here is just one of those examples, a different species there of a wasp-type critter. And here is a frog that was trapped in amber. And it has been CT'd so that way the specimen can be studied in great detail without causing any damage to it.
We're fucking crazy.
So this here
My
you see
dad's
the
high
jaw
risk.
and the forelimbs and part of the spine of the frog. And then here's a close-up showing exquisite detail of its architecture, and then also some of the limb extremity anatomy as well. So here is an article of, highlighting these various specimens here from Chinese scientists working on the specimens from Myanmar, or as it used to be known, Burma, and showing that there's this wide age range back to, in their example, dating it to 99 million years ago. This has, however, caused a problem politically because of the very severe conditions under which the specimens are mined. There are issues of how the laborers are treated. And this has led to scientists around the world saying we should not publish any articles done on specimens from Myanmar, from Burma, because of the way those people are treated. So the journals are encouraged to not publish articles on that work. Here you see the map now having an additional dot indicating the northern shore there of Australia. And this is the most recent location where amber has just been showing up in the last couple decades. And so here's a picture taken of the shore there, showing what has been washed up from the ocean. One of the most unusual specimens is this type of beetle that kind of looks like it came out of Star Wars or Star Trek or something because of that very odd shape that it has there, not identified or seen previously anywhere, as well as other specimens that might seem not so exotic. So there's quite a variety there. The Baltic source of amber is this tip of the coast there in Sweden, where in Denmark almost come together. And then from that place, it was transported and radiated to other shores on the Baltic coasts there, including this part of what is the Russian Federation. And this is a interesting political situation where that area outlined in red is still part of the Russian Federation, but because of the dissolution of the Soviet Union, which then included Estonia, Latvia, Lithuania, Belarus, and other constituent republics further south, it is no longer contiguous because those countries achieved their complete independence. So here is a close-up of this enclave, and you see there, Kaliningrad is the modern name for the city there, the Russian name for it. And then on the coast, you see Yantarny in the red rectangle. And here's a close-up giving a more detail of the geography of the area. And so looking west from that shore, this photo I took when I was there, I'd say it's about close to about 12 or 15 years ago now, and was taken there as part of my mission trip to teach creation science in places in that region. And so I got to actually photograph where the mining of amber was taking place there. And you see that it's a pretty thick layer, and it's called blue earth. And you can see why in this shot. It better shows the coloration there from which the amber is mined. And this is a big operation using large equipment, large scale equipment here, which is moved around as the area under the equipment is next scheduled to be mined. And so you can see how these things are up on these very large supports underneath, and then huge cranes pick these things up and move them around from place to place as they shift the location of the excavations. And so they use these derricks and the conveyor belts then to move the material that's mined out of the region here. And so you can see it's a very extensive, thick layer of the blue earth. Well, there's at the observation point there, they have this little pit that they've arranged for children. They salt it with specimens of amber so the children can dig up their own little piece of amber there. And then in the neighboring town of Yantar, which is the Russian word for amber, so the name of the town is Amber, there's this museum there with a collection of remarkable pieces and things made from amber. Well, at this point, I want to give you some very important history that relates to this region and to amber. And so in Europe in the year 1700, you see the areas of pink surrounding a great part of the Baltic Sea, including a small part on the southern coast there, which is now has the oval, pink oval around it. And so because of this, the Baltic was referred to as a Swedish sea, S-E-A, sea, because of so much of the coastline being controlled by Sweden. And you see right next to that enclave on the south shore is Brandenburg, along with Prussia. So they were together under the rule of Frederick I of Brandenburg. And he was not crazy about the idea of the Swedes controlling that coast right there, right on his border. So he approached Peter the Great, Peter I of Russia, and said, how about joining me in a happy little war against Sweden so we can both get more of the Baltic Sea coast? And Peter was all for that because he wanted to have a seaport on the Baltic Sea. He wanted to have his, quote, window on the west. And he had spent time before becoming Tsar in the Netherlands, and what's labeled there is the Dutch Republic at the far right, left side of the slide, firsthand learning a lot about seamanship and seagoing vessels. And so he wanted to be able to employ that in his window on the west on the Baltic. So he joined in with Frederick I. They had their happy little war and indeed conquered the territories that they wished to obtain. And so there you see where the red dot has moved in at the top there, became the newly located capital of Russia, known as Petersburg, at the time, St. Petersburg. And so he shifted the capital from Moscow to this newly built city, which was built on marshland that had to be reclaimed. So canals were dug, land was drained, and he built his new capital there. And so meanwhile, back in Berlin, this was the Charlottenburg Palace that Frederick I lived in, and he had in this palace a room totally lined with amber that had been acquired from the Baltic region. And he, as a present, as thanks to Peter for his cooperation, he gave this amber room to Peter the Great. So in this newly constructed palace there in this new city of St. Petersburg, where the Neva River empties into the Baltic Sea, his window on the west, he built this palace. It's called the Winter Palace because this is where the Tsars would live in the wintertime. And he had his amber room. So there you see the eastern end of the Baltic Sea. And so there now, where you see that red balloon-shaped marker is where St. Petersburg was located. And then south of St. Petersburg, Catherine, a subsequent ruler of Russia, a couple generations after Peter, built her big palace for a summer residence and built this humble little dwelling, as you see here. And she liked the amber room so much she had it moved from the Winter Palace in St. Petersburg to here at a place called the Tsar's Village, Tsarskoye Selo, where the Catherine Palace was built, as you see pictured here. And in here, then, is this amber room, brightly lit with the lights, which, of course, in those days were candles. So the lights are today still shown as candles, even though they're electric. So that's the history of that region that amber played a very big part of. Then World War II rolls around, And this is what was done to that Catherine Palace. Well, when it became obvious that after the German army started its roll eastward, the caretakers, curators of the palace did everything they could to salvage, take out from these structures, whether it be here or the other summer palace that Peter had built on the Baltic Sea, closer to St. Petersburg, to take all the things that they could that were removable. So Catherine had told them, try and save all the amber, take the amber out of this palace. Well, they couldn't because it was too brittle by then. And so after the unsuccessful attempt to try and remove the amber, they papered over it with wallpaper in an attempt to hide it. But that was a feeble attempt. It was not successful. And so the Germans removed all of the amber from the room, boxed it up in some 30 wooden crates, and then shipped it out before totally destroying this building. And so here you see what happened to the interior of the building. So everything was gone, either from the Russians removing it for safekeeping or the Germans destroying what was left behind. So here back in this enclave, you have to know that at the time of the beginning of World War I, some of this was still Prussian territory, meaning German-speaking peoples, and the city was still known as Königsberg at this time, before being renamed Kaliningrad. And this was the structure, it was known Königsberg, there it is in German, and this was the structure there in that city to which the amber was taken in those 30 crates. And of course you see all the Nazi symbols and everything all over the place. But as this region was bombed as the war proceeded, this was the building reduced to this kind of a rubble. Same thing as what happened to the Catherine Palace. So then the question is, what happened with all the amber that had been taken there? And the answer is, it's not known to this day what happened to all of it, or actually almost all of it. Various theories are the bombing destroyed it. It was put into a ship to take it to Germany from Königsberg here, across the Baltic, but that the ship was sunk and lost. Or it made it to Germany and the stuff was successfully hidden to the point where it's still not known, except maybe a few secret people, where it's at. So nobody really knows. A couple of items from the amber room were eventually located at the turn of the century and returned to Russia, hoping that Russia would then return to Germany things that were looted from that country at the end of the war. So all sorts of mysteries still persist. So in subsequent decades, bit by bit, as money became available, the Russians rebuilt these palaces that were so destroyed and restored them, and at great cost built the modern version of the amber room today. So that's what these photos of. There's from the reconstruction that was done since World War II at tremendous cost. So we'll turn now to where did all this amber come from? How did it form and when? So here's that slide I showed you before that according to the secular scientists, it was thought to happen over this extended time period of some 300 million years. And encased, we see various specimens here from plant and animal life. And so one of the fellows who's doing research currently says it's difficult to date amber directly. So what they try to do is date it by dating the rock layers in which the amber is found. We've heard this story before in other settings. And so here is the reference for that quote, if someone wants to look it up, as you will have these slides. And so what are the dates? What are the mechanism? this is, these are very important questions. And the common thought given to us is that amber formed from resin because of a wound inflicted somehow, and that it would slowly flow, but things would end up being trapped in the amber in the location where it was forming as the wound was being healed by the tree. Well, okay, how does this work? Well, we can understand that some plant material might be blown onto the amber by some vigorous winds, including this extinct flower here, trapped in amber. We could understand that thinking that maybe these terrestrial insects living on land would crawl onto the tree and get stuck to the amber and then trapped by subsequent flow, including this mite, this very small organism, which comes from this bird specimen. And even bacteria inside the flea, the mite, the flea, which has actually been able to be identified as the very same species that caused the Great Plague, the bubonic plague of the Middle Ages. It's very interesting that even though this is supposedly hundreds of millions of years old, we can still identify the organisms, the microorganisms inside the flea, inside the amber. Isn't that crazy? Makes you wonder, are those hundreds of millions of years real? Well, here you even have larger things such as a feather, significantly sized feather, and trapped in the feather is this tick-like organism that you see magnified in the upper right part of the image. Totally identifiable as to species. Okay, what about these larger animals, terrestrial animals? Here we see scorpion in amber, or a frog in amber, or part of a salamander in amber, or an entire salamander in amber. This makes you question about this flow theory on the tree at the site of a wound on the standing living tree. How about termites that are found with organisms in the gut of the termites that are identifiable as the same as organisms in termites today? This gets pretty remarkable, I think, that there are all these generations that have passed in subsequent 20 million years, according to evolutionary dates, yet no evolution. It's exactly the same thing. You know, that should knock your socks off. So here we are, dated at 100 million years, and what are we looking at? We're looking at part of the gecko here. Well, okay, that's fine for terrestrial organisms, maybe, it's a stretch. But what about freshwater organisms found in amber? How did they get up onto those trees? This becomes too much of a stretch. So we have these things such as this water beetle, or a water strider, freshwater riffle beetle, freshwater snail. How did that snail get up on the tree? And now even a greater problem is, what about these marine organisms found in amber? So barnacles, which, you know, fasten to rocks or to bottoms of ships or to piers, you know, how did these things end up in amber? How about isopods, which are very small crustaceans related to crabs and shrimp? How about crabs themselves? How about marine snails? How about red algae that lives on the bottom of the seafloor? How did all of these things get trapped in amber? Now, how about a trilobite? Now, that should really knock your socks off, since these things have been supposedly extinct for extremely long periods of time. They're found very at the bottom of the geological column. How about ammonites? As well as all these other marine critters. Just amazing. All in the same specimen, and some critters that are not marine critters, all together in the same specimen. Remarkably beyond belief are air bubbles trapped in amber. So how did that happen if these things are just from trees standing up? Well, some of the evolutionists try to explain away some of these bubbles as saying those are a result of gas from decomposition of these animals forming the bubbles in the amber. But here we have, where that's not even a possibility, multiple bubbles in an amber specimen with no animals. And analysis of some 400 specimens of air bubbles in amber show that the oxygen percentage is in the 32 to 36% range, which is significantly higher, up to 50% higher than atmospheric oxygen, which we know is 21% oxygen. What's super remarkable is this air bubble trapped in water, trapped in amber. So with this comparison of these two photos shows that with a shift of the piece of amber so that the orientation of the water bubble shifts, the air bubble in the water moves. Just like what you see with a carpenter's level. As you shift the position of the level, the air bubble in the fluid in the carpenter's level changes to indicate, the relative position. This is beyond belief. This can't happen in a tree standing up on land. And an analysis here, refers about this gas composition, and they quote, assume ancient air there in the title. And so I, would repeat the caution given to us by Jonathan Sarfati, Dr. Sarfati, saying that even though these measurements of oxygen levels of 32 to 36% in these bubbles don't necessarily mean that the pre-flood atmosphere had that higher level of oxygen. It may very well be the case, but he thinks that that's a bad assumption to make. He says it's a possibility, but not something we can assume to be fact, since we weren't there to see exactly what the conditions were in the atmosphere prior to the flood, but it's a possibility. This slide here I put to emphasize the amount of carbon that is precipitated out into rock these days that would have been in the biomass, in other words, in the plant and animal material prior to the flood. And you see here that there's 20 million units, and it's important to pay attention to the footnote where each unit is a billion tons of carbon. So we're talking 20 million times billions of tons of carbon. Tremendous amount of carbon that was in circulation in the biomass, in the plant and animal material, that now is precipitated out into rock in its form as carbonate. Also taking into account looking at the other places where there would be carbon locked up, whether it be in oil or in peat, partially, this decomposed organic material that can burn. So there's a lot of carbon that was in circulation that is not now. So the speculation of putting all this together is that the atmospheric pressure was higher prior to the flood, including much more increased amounts of oxygen and carbon dioxide, raising that barometric pressure, allowing these very large organisms, the pterodactyls or pteranodons, or even these 30-inch wingspan dragonflies, and then these extremely large flying critters the size of giraffes, being able to fly by having enough lift with a much denser atmosphere, much higher barometric pressure. So this is speculation, but it is a distinct possibility. So returning back to the bubbles you see here, how do we explain these things in the amber? Well, these two secular scientists may have an answer that puts a lot of this together. The one fellow here from the museum in Berlin and the other one in Florida did some work in the swamps there in Florida, where there was some privately owned land where they could purposefully injure these cypress trees. And so here is the saw here where there's some wounds put into the cypress trees. And then they observed what happened with the resin that flowed from these wounds. And they noted that it would flow into the water, and with the resin being in the water there, rather than exposed to the oxygen in the atmosphere, it did not solidify rapidly. It was much more fluid in its state, and was also stickier longer underwater, and therefore being in a much more favorable position to be able to trap anything that was in the water. Could be from a freshwater source, a marine source, or a terrestrial source. And that there was much more time allowed for the resins to flow to totally entrap these larger specimens from whatever their source would be. So then putting this all together, we have to think of some kind of event that would destroy trees on an extremely large scale with huge amounts of resin being liberated in a marine water environment, which would include marine, freshwater, and terrestrial sources for the animal and plant specimens. And so here you just see what happened with the results of a hurricane in Florida on a relatively local scale compared to something much more drastic. So in their study, their resin actually did not solidify. It remained in a more liquid state, but they said with being buried by overlying layers of sediment, then that would certainly give rise to the possibility or even probability of solidification being accelerated. And so can you think of a place where you could see layers of sediment that would provide that weight and even heat to cause the resin to solidify into amber after trapping these organisms? And what place can you think of where you can see all of these layers? I won't be able to hear your answer, so I'll go ahead and just insert it here, and that would be our good old Grand Canyon here in the state of Arizona, where you can see these layers, showing where this would be able to happen around the world with a worldwide process. And so they say, given enough protection by layers of sediment, the amber could survive intact for millions of years, given the evolutionary time frame, since they are secular scientists. So the event that would put all of this together would be, of course, as we think of it, the Genesis flood, Noah's flood, a worldwide event. And so here is some of the work from the Institute of Creation Research, showing how, analyzing the various layers, continent by continent, they think they have a pretty good idea of the ebbing and flowing of the waters as they became deeper and deeper, accumulating around the world, shifting stuff around. And so in a particular location, the water would be up at a higher level, then go down and be up at a higher level and go down. And then finally there would be total immersion underwater. So that's what these maps are I'm trying to indicate here, taken from the work that they're doing. They've done the two American continents and Europe, and now they're working on Africa and Asia. So water immersion is necessary for amber to have time to flow and totally encase these specimens, so the organisms, plant and animal. to capture the marine and fresh water as well as the terrestrial organisms to occur on a worldwide basis. And so the simplest explanation, of course, is given to us in chapter 7 of Genesis here. Tremendous amounts of water, and not only water, but lava being these fountains of the great deep. So here's an illustration from Walt Brown's book in the beginning with the artist's impression of how when the crust was ruptured, that the water would be coming out from an extremely high pressure from within the crust, and then beginning the process of inundating the land. And then the water continuing to rise, taking time to be able to do so to completely cover everything, and then remaining high, so that for the first 5 months of the flood year, the inundation and the water staying at that high level, covering everything. So therefore, the ark was then able to float anywhere in the world with a minimum of 23.5 feet under it at the highest point of the land underneath the waters. And I'd say minimum because the cubit could have been longer than the standard cubit. It could have been a royal cubit, at least 10% longer. Well, it's very interesting that in the last couple decades, there are now several quotes of secular scientists saying these things about how much water was upon the earth. An ocean world. Evolutionists admit that this planet could be covered almost entirely by water. We're talking about a time when, if you were looking at the earth from space, you would hardly see any landmass at all. It would have almost been an ocean world. So this was published in 2009. 100% ocean world. However, some recent studies suggest that water may have covered Earth's entire surface for some 200 million years before the continents emerged. This is published in 2013. A new theory has emerged that almost all of the Earth has been covered with water millions of years ago, 2016. 4 billion years ago, Earth was covered by a watery sludge, 2017. Early Earth was covered in a global ocean and had no mountains, also in 2017. These are all secular sources. This is astounding, and yet we never hear about this. So isn't it amazing how in this area, as well as so many others, that they keep getting closer and closer to the truth? It's astounding. But the public never hears about this. Well, indeed, we have the history in Genesis chapter 7 showing that indeed the entire planet was covered with water, and all living critters that did not have the breath of life through nostrils, not on the ark, died. So that would be birds, mammals, and reptiles. Of course, fish don't have nostrils, and the amphibians in the phase of their life also not having nostrils, so they could survive in the waters, not on the ark, if they didn't get buried. So here we have a modern photograph of a marine volcano underwater. Tremendously large marine landslides, I guess you call it a marine slide, over several degrees of the Earth's circumference, at least 4 degrees. So we're talking a big distance there. Tsunamis, going all over the place, back and forth with no land to interrupt them from these marine landslides. And so everything, again, not on the ark, destroyed. Just give us emphasis here, repetition, making sure we understand this. And then also storms, very violent, very huge storms. This is a picture from Katrina, small portion of that. It was calculated that the hurricanes ravaging the ocean during the year of the flood were 100 times larger than the modern hurricanes today. So this had to definitely bring home this concept of the ark being the protection, the refuge for the organisms, as the ark is the picture of Christ being our refuge as well. And so from Genesis here, I wanted to emphasize the business of the instructions given to Noah, chapter 6, about making the ark and covering it outside with pitch. In other words, some sticky substance, maybe from a petroleum product, maybe from plant products, as in resins from plants. We don't know for sure. And here in Exodus, also dealing with Moses, the same words for ark and for pitch. And so here we see an artist's conception of the ark that Moses was put in and then put onto the Nile River and then taken up by Pharaoh's daughter. And so here we have this reference here of Christ saying to the disciples, don't worry about your life, what you'll eat, about your body, what you will put on. In other words, clothing and or jewelry. So that's the link to the reference to jewelry made from amber. And some of it is so exquisitely beautiful, it's phenomenal, whether it is with or without the inclusions. So you see here a wide variety of things made from amber. But the true jewels that we have in the arks of Noah and Moses and Christ are the souls that they contain. Though those who put their faith in Christ as their creator, savior, and redeemer. So that takes us from amber itself to this amber colored substance we call honey. And these jars represent specimens of honey here produced here in Arizona. Very tasty. And so the question is, how are bees in amber causing headaches for evolutionists? So that's what ties these two subjects together. And it says here that these bee specimens in amber have changed very little in the last 80 million years. Well, of course, we understand that that time is not existent. And as the flowering trees supplying the resin for the amber specimens, causing a 100 million year problem, these fossilized, mineralized combs from bees also cause a 100 million year problem for the evolutionary dating, because they are found in rock layers that appear 100 million years prior to the supposed time of the appearance of flowering plants. Well, we know that bees need the flowering plants for their sources of nectar and pollen, and the plants need the bees for pollination. So this is another version of that 100 million problem. Instead of being the amber itself from the tree sources of resin, it's the honeycomb here, the combs from animals that require flowering plants as well. So this bee is saying to us, I mean business. So let's get down to business and ask this first question: how do bees use their senses to find nectar and pollen? Well, they use their antennae, which can be for a sense of smell, as we think of it, as well as for touch. Mouth parts as well, in terms of being able to have some ability. And then there's the part of the foot called the tarsus. And you see here the human foot and ankle, and the colored portions represent the bones that make up what we call the tarsus. And so the analogy there is that's the same part of the foot of the bee. And on the tarsus of the bee are these very special cells which give them a sense of smell. So these cells, sheath cells, are used to be able to pick up molecules in the air that are providing that sense of smell, of odors. And here is a micrograph from electron microscope showing these structures, these sheath cells with the nerve ability to be sending the pickup of these molecules onto the bee's brain to know that it's smelling whatever these molecules provide. Well, here you see a type of plant called oleander, which grows in the desert areas. We have it in Arizona. I've seen it in California as well, and other warm climates. And it has this particular molecule called oleandrin in the nectar. And this is a toxin. This is not just in the nectar, it's also in the petals, in the leaves, in the stems of the plant. And so when the bees have other sources, they don't take this, they avoid it as much as, until they absolutely have to take it. They know it is toxic. And so people who eat any part of this plant become poisoned. And the mechanism is this acts as the same as the drug digitalis, same effect on the human heart. And so when we've had to treat patients, especially children who eat part of an oleander plant, or adults who use the branch from the oleander to put a marshmallow on to roast, or a hot dog on to roast over a fire, they end up getting toxicity. And so we treat them as if this is a digitalis toxicity, and are able to help them out with their problem. So the bees are able to understand what is a sweet thing. They can pick up sweet. They don't pick up the bitter per se, but they stay away from it if they can all do so. Well, another fascinating thing is some flowers have these literal physical landing strips for the bees to land on to be able to access the nectar. And bees have these special sensory organs on the forehead. You see in the red circles, one is called an ocellum, ocelli plural. And these have just one lens, one curved lens. And the only purpose for these is to be able to detect motion, not to actually see an image of something, but just to detect motion. And that's so that the bee can know that, like, for example, if there's a bird coming at it to try and eat it for lunch, to evade the bird, for example. So here is the anatomy of the compound eyes outlined in yellow, one on each side of the head of the bee. And these have these very long structures called ommatidium that have one lens per ommatidium, but there's thousands of these that make up the compound eye that you see outlined in yellow there. And so here is the field of vision of the human being with a total of 190 degrees. So we can see just a little more than halfway around. But in comparison for the bee, they see 280 degrees because their compound eyes are located on the sides of the head rather than in the front as it is with us. So they have a tremendous range field of vision there. And so with those large eyes on the sides of their head, you see here with magnification, you can see these compound arrangements with these thousands of ommatidia here, with increasing magnification with an electron microscope to where you're seeing the individual ones here. And these have different color ranges that they pick up, the different ommatidia. And you see these little tiny spikes coming out, which we'll get to in a little bit here. So here is how the ommatidia are constructed with the rod. Rhabdom means rod, and this is then how the light is perceived with each particular small lens here, with the signal picked up by it transmitted through the rhabdom and then into the nerve, the axons that combine to form the optic nerve going to the bee's brain. So these are the color ranges that the bees can see. They don't see red. They don't have the ability to pick up red, but they do have the ability to see ultraviolet that we don't have. So their color range of vision is shifted to the blue end with the ultraviolet extension minus the red end. Well, this is important because this is how they see a flower compared to how we see a flower on the left. So visible light on the left, ultraviolet light on the right. So they see patterns that we don't see, that are not visible for us. And here you see other examples of visible versus ultraviolet. And here as well. And in these, the left side visible, the right side ultraviolet light. So it's fascinating. Their world looks very different than ours. And we'll get back to this business of ultraviolet light in just a bit. Well, another amazing thing is plants can sense the vibration produced by the beating of the wings of the bees. And so with the sound of a bee's wings flying, recorded and then played back, plants can be stimulated by receiving that sonic message to increase their flow of nectar and to increase the sweetness of the nectar by 20% in the course of 3 minutes. Isn't that astounding? That is just absolutely amazing. So the plants sense the vibration caused by the flapping, the beating of the bee's wings, stimulating more nectar, richer nectar. So I wouldn't call it hearing per se, but it is in a sense that because of sensing the vibration. Well, not only is the nectar flow increased and made more sweet, the plants will release pollen in response to the vibrations sensed by the beating of the bee's wings. And what makes that even more interesting is the static electricity that is part of the pollen causes the pollen to actually jump onto the bee's body hair. As you see here, this bee is plastered with pollen. Well, how do the bees know where to go? So here you see, putting a lot of this information together, you see here that the angle of the lines formed by the line from the sun's position to the hive and from the hive to the source of nectar or pollen, in this case, forms a 30-degree angle. So the bee will go back to the hive and reproducing that 30-degree angle from vertical line of gravity. This waggle dance is then sensed by other worker bees inside the hive and receive the information on where to go to find the nectar or the pollen. The angle reproduces the the direction and the speed with which the bee vibrates the abdomen indicates the distance. It's absolutely astounding. So here you see this 30-degree angle of the bee, and this again is in the dark in the hive, and the other bees there are picking up on this direction, the 30-degree direction from vertical, and how fast the abdomen is being waggled for distance. So we will get to the next slide here, and then watch the bee there do the dance, go back around and repeat it. And so you can see that this angle is away from the sun, not quite 180 degrees. So it goes to another area so more workers can pick up on the signal. And then we'll repeat the dance again. Same angle as before. Goes back up, comes back down. So that's not a real fast vibration there. Well, at one time it wasn't so fast. But that speed of the vibration indicates the distance. And also they may pass out samples of nectar so that they have that smell. Isn't that amazing how this all just happened?
Yeah.
All right. Whoops, I want to move on. Well, when the sun is shining, that makes it easy for them to reproduce that angle. When the sun is not shining because it's cloudy, then that makes it tougher, but they still have the ability. So here on a cloudy day, they can still sense that angle from the sun because of the polarization of the sunlight. And so going back to these ommatidia, in the left-hand part of the diagram, you see how there are hundreds, thousands of them in an eye. In the right part of the diagram, you see that they are arranged in these groupings that are 90 degrees to each other. And that's how they're able to sense the polarization of the light. So even with the clouds and inability to see the sun directly, they can still figure out the correct angle and reproduce that. Now, when I was a teenager and in college, my dad and I kept bees, and we can tell you from personal experience that on a cloudy day, they are more grouchy and are more likely to sting you than on a clear day because it's a bit tougher with the sun blocked by the clouds, but they can still do it. So I made reference to those little spikes you saw on that electron micrograph of the eye, the compound eye. And so the question arises, do bees have hairy eyeballs? And the answer there is yes, indeed they do. So then the question is, why would the bee have a hairy eyeball? And it is thought that with this, they're able to detect the effect of crosswinds, throwing them off course, so they're able to compensate for that and stay on course to get to where they're trying to go. Another very fascinating thing is what's called the traveling salesman problem. And so the question is, you have these multitude of flowers for either nectar or pollen. How do you go from one to another in the most efficient manner without unnecessary backtracking? So because a bumblebee is larger than a honeybee, they used a bumblebee and they were able to put a radio transmission tracker on the bumblebee. It was so miniaturized, and the bumblebee, time after time, correctly solved the problem with the most efficient path from flower to flower, confirmed by computer programs programmed to solve the same problem. Just absolutely astounding how these tiny little critters can do that. Well, not only that, they can understand the concept of zero. How in the world did we figure that out? Well, the bees were trained they were offered, samples of nectar, and they were trained to do it in sequence so that the number of dots from one occasion to the next was decreased from, you can see here, the larger number of dots, 5 being the largest number there, down to finally where they took it down to no dots. And the bees would consistently be able to comprehend the absence of the dot and go to the place where there was no dot and the nectar was there. Fascinating. Fascinating, beyond belief. So how do they bring the pollen back? So here is a lateral diagram of the anatomy of the bee. And I've highlighted the upper part of the hind leg. And so here you see that pollen sac on that upper part of the hind leg filled with red pollen from these flowers here. I took this photo when I was in the Ukraine on a mission trip a couple of years ago. And then here you see another example where it's even more stuffed with pollen, yellow pollen in this case, from these particular flowers. And you see here, as I mentioned, the electrostatic charge where the bee gets covered as the flower releases the pollen upon sensing the vibration of the bee's wings. And so it's very interesting that the size of the pollen grains is just the right size to fit in between the hairs on the bee's body for maximum capture. And here is showing the electron micrograph of the pollen grains and the body hair of the bee. And the bee is equipped with the comb on the leg to be able to comb out the pollen and stuff it into those sacs, those pollen sacs. So the bee is combing itself to take the pollen off the various parts and stuff it in. And so here you see that leg extended there over the top of the head to comb off the pollen and put it into the sac. I would like to be as dexterous as the bee is in order to be able to put my leg up over my head like that. I certainly can't do that. So here you see these extremely stuffed pollen sacs on the hind legs there, as well as those hairs able to carry even more pollen that were not combed out as the animals fly. So you can see this critter is pretty heavily laden down here with the pollen. So when they get back to the hive, they empty out the sacs, comb themselves, and stuff the pollen quite tightly into the cells here for storage, and then they cap it. So the pollen is their source of protein. The nectar is their source of carbohydrate. The pollen is their source of protein. And there is a certain amount of fats in both the pollens and in the nectar. There is a little bit of fat in the nectar as well. So how do the honeybees bring back the nectar? Well, here you see a lateral view, the gastrointestinal tract laid out in green. And you see the label on the far left there of this sucking pump. And then you see what's labeled as the honey stomach, the first stomach. Its proper name really is the communal stomach. I think that's the more professional term. And then the rest of the GI tract that's used for the bee's own needs. So the nectar is transported in that communal stomach and then regurgitated back upon arrival in the hive into the cells. So here you see a close-up photo of that sucking pump, and you can see it has several small appendages at the end. It's not just a soda straw that's cut off cleanly at the end. It has these several small appendages that are used. So here you see where the nectar has been regurgitated back into the cells in the hive, and then you see some of it's capped, and we'll talk about that in a second. So what's the difference between the nectar and honey? It's a matter of concentration. So here the bees are attaching themselves with the hooks on their feet to the wood at the entrance of the hive and are fanning madly. So they're, quote, flying in place, collectively making quite an air current to speed up the evaporation process so that when the honey is at the right concentration, they then cap those cells with wax, cover up, and seal it off for later need during the winter months. And they know instinctively at what point it is concentrated appropriately before capping off the cell. But so the question is, where do they get the wax? Well, I'm going to answer a different question, but where do they get the wax from? First, we're going to answer this question of propolis, P-R-O-P-O-L-I-S, propolis, which literally means in Latin before the city, the front of the city. So here you see the entrance to a hive, which has been greatly narrowed down by the bees placing propolis there. And there's two major reasons for them doing this. One is to make the entrance much smaller, to make it easier to defend against invaders, honey stealers, such as a wasp, which would come and steal honey, or possibly bees from a different hive. The other is then to better preserve the heat in the hive in the wintertime, especially for bees who are in very cold climates, like in Minnesota or other places further north like that. So here you see a comparison of March 2nd and April 9th, where as it warms up, they're removing more of the propolis to get better ventilation and keep the hive at the same temperature without it overheating. Where do they get it from? To answer the question posed, well, a major component of propolis is the resin from buds of deciduous trees, specifically deciduous trees. And the contents of the resin, chemically, the molecular makeup of the resin varies with the species of the tree. This is a major component, and this resin is, quite sticky. And propolis is phenomenally sticky. And the bees use it not only to control the size of the opening, the entrance to the hive, they also coat the inside of the hive as well as the honeycombs themselves. So as the honeycombs get older, they age with more propolis put on them. So they become more yellow from the propolis. And the bees do this because of what is contained in the propolis, besides its ability to stick things together and to glue things down. So here you see a listing of the contents of propolis. So the resin from the buds of the deciduous trees make up about half of its total volume. And you see here that pollen, they mix pollen in with it for 5% and wax 30%. So some 85% is pollen, wax, and resin. Oils that they collect are another 10%. And then other organic compounds, the remaining 5%. Now you see here highlighted in yellow what's called flavonoids and in vitamins B1 and B2, because those are from the pollen itself. And then these minerals are from the resin, so they're color-coded with the word resin, all these various minerals. And then there's all sorts of these enzymes, and you see that these are enzymes that break down molecules, such as here adenosine triphosphatase, which breaks down ATP, as well as these other enzymes that break down organic molecules. So all of these things are in propolis. It's amazingly rich substance, which has not just the resin, but these other things as well. And it's found out upon a lot of extensive work by scientists all around the world that these are the properties of propolis due to the contents that I showed you on the previous slide. So they have antibacterial, antifungal, antiviral properties. So that's why they coat everything in the hive, so the hive can not receive infections, or at least have reduced chance of infection. And then it has these other properties which are very beneficial to people: anti-allergy, antioxidant, anti-carcinogenic, causing cancer, mutagenic, meaning preventing mutations in DNA, and anti-inflammatory. So it's amazing. So these things have medicinal qualities as well. Well, back to the question I asked you earlier, where did the wax itself come from? So where in the heck do they get the wax? Do they go out and collect it somewhere like they do the resin from the trees for the propolis? The answer is no, they manufacture it. They have these glands, eight glands, 4 pairs in the underside of the abdomen that produce the wax. How do they do this? Well, they have in these organs, these extremely long molecules that are chains of carbon and hydrogen, and have this grouping on the end that's called a carboxyl group. So these are called carboxylic acids. And they also have these very long chain molecules that are all carbon and hydrogen as well, except for the one end, which is a hydroxyl group, which makes it an alcohol. So in that gland, these two molecules are combined, and I highlighted the one hydrogen in blue and the other hydrogen and oxygen in blue because those are removed by an enzyme. These two long molecules are combined at these points into this very long molecule, and all of those angles are represent carbons with hydrogens. And you see there the remaining two oxygens in white that are in the middle of that molecule there. And then you see the blue water molecule resulting from the hydrogen from the one and the oxygen hydrogen from the other being combined to make a water molecule. So by dehydration condensation, these extremely long wax molecules are made in those organs on the underside of the abdomen. So those little platelets of wax are used then to form the honeycombs to make the cells that make up the hive. And it takes 9 pounds of honey in order to produce 1 pound of beeswax.
Wow.
9 pounds of honey they ingest in order to make 1 pound of beeswax. But Your Majesty, what's the point of being busy bees if the so-called beekeeper always steals our honey? Well, good question. Obviously, it's just a joke. So here you see a bee using that sucking pump to soak up water, to put water in its communal stomach. Well, why would it do that? Well, in the hot climates, they need to keep that temperature in the hive from rising too high, just as in the winter, they need to keep it from going too low. And so they bring the water back to the hive, deposit it in the hive, and then do their fanning thing, this time not only to evaporate the water out of the nectar, but also to cool down the hive. So here in Arizona, they do this a lot, and wherever you see a dripping faucet or a water source, you'll see bees madly getting water so they can take it back to the hive and cool down the hive when it's really hot here. So then the question arises, what does honey actually cost to produce? Well, this is an older cartoon. I couldn't find a date with it, but at the time it was drawn, it said that a jar of honey would cost 182,000 bucks. Well, I did some real math for current times. And so to produce 1 pound of honey, here's what's involved. And this has been very carefully studied. So these are, pretty accurate numbers here. 556 worker bees flying at a top speed of 15 miles per hour, fly 4,600 miles, visiting 2 million flowers. Flying at that rate, they visit a flower for 12.5 seconds. So it's 312 hours of flight time. Imbibing 7,000 nectar for 7,000 hours. Having to condense that nectar down to 9 pounds of honey to produce 1 pound of comb. Well, a gallon of water weighs 8 pounds, but a gallon of honey weighs 12 pounds. So that means they have to evaporate out a third of the volume of the nectar to get the honey. So the work of evaporation, cooling, guarding the hive, and human labor are excluded from this calculation. So it comes out to, last year in Arizona, 2019, at a minimum wage of 11 bucks an hour, $804,100 per pound. This year, with the minimum wage now being 12 bucks an hour, it now is $877,200 per pound. So enjoy that honey on your toast. There's a special, are several special sets of glands in the bee's body, in the heads and in the chest, the thorax. This one particular set is called the hypopharyngeal glands, and they produce enzymes that break down the sugar molecule in honey called sucrose. Sucrose is a molecule that has two sugar molecules making it up. One of them glucose, the other one fructose. So when that bond connecting glucose and fructose is broken, the sucrose is converted in these two separate monosaccharides, one sugar molecule. Well, another name for sucrase, which is the name of this enzyme breaking down sucrose, is invertase. You'll see it referred to as that. Well, other substances produced by the same gland besides the sucrase or invertase is called gluconic acid, and the reason for the gluconic acid is to lower the acid-base balance, make it more acidic, to lower the pH, to make it more acidic. And also found in these secretions from this gland is hydrogen peroxide, H2O2. The properties for this is that the acidity helps prevent growth of bacteria, microorganisms, and other microorganisms, as well as bacteria. And the hydrogen peroxide oxidizes the microorganisms. So that's why honey can sit on a shelf forever and not spoil, because of the acidity and these molecules such as hydrogen peroxide. Well, there are other glands as well that secrete other things, as you see here, thoracic glands and head glands. And here is a listing of many of the things that have been identified so far in these secretions. And I'm going to give you some idea here with some color coding that these in this very pale blue, number 9, 12, and 19, are enzymes that break up the proteins that are in the nectar into the individual amino acids. These that are color-coded in yellow break down these other complex carbohydrates that are in the complex sugars that are in the nectar in addition to the sucrose. In green here, this enzyme lipase breaks down the fats that are in the nectar. And then here is a very interesting one that's in a more of a tan color that breaks down chitin. And you're saying, what in the heck is chitin? Well, chitin is the stuff in polymer form, in long chain form, that makes up the shells of beetles, makes them crunchy, and the shells of crustaceans and these insects like a grasshopper. So here is the unit. It's a sugar molecule with this other thing called N-acetyl group that is on the sugar, N-acetyl glucose, and N-acetyl glucosamine is its full name. And so chains of these things are what make up chitin. So you're asking yourself, well, why are we talking about an enzyme that breaks down chitin if chitin is what makes up the shells of beetles and insects and crustaceans? Well, it's because this enzyme with this name breaks down this same polymer that's in the cell wall of these certain types of fungus. Well, why are we worrying about fungus? Well, it's because this fungus will infect plants such as the wheat plant and then will blow into the nectar and pollen of the plants that the bees harvest. So in other words, this fungus could then infect the honey, the nectar and the resultant honey. So God provided for this enzyme to break down the chitin so that the fungus does not survive and the honey can still be stored forever without being spoiled by the fungus, as well as by bacteria, being protected against those things as well. Well, what about robbers who want to come in and steal honey? And this happens in the insect world. How do the bees deal with that? So here you see that there is a wasp that does come in and try to steal honey. Well, the wasp has been immobilized, so this film can be made. And this shows that the bees will then collect and form a ball. So this is called balling the wasp. And you see these abdomens madly vibrating here. Let's see if we can show this again. Let's go back and go forward so that you can appreciate this. And they're madly vibrating their abdomens to generate more heat, to burn up fuel, to generate more heat, to raise the body temperature of this wasp that would be invading the hive. And so here you see infrared picture of this doing this. Well, how do they know just how much to do this to deal with the temperature? So here's a scale here showing you that the honeybees, the European honeybees, can handle temperatures until they get to 103 to 100 or 104, 124 or 5. The Asian honeybees can handle the temperatures until 123, but the wasps are killed somewhere between 110 and 116 degrees Fahrenheit. So the bees can sense how to do this up to that range to kill the wasp, but not enough to kill themselves. Absolutely astounding. Well, as we talk about these various sets of glands, here's another set that hadn't been mentioned yet called the hypopharyngeal under the pharynx gland, because that's where it opens into the GI tract. And this is where royal jelly is produced. And it's called royal jelly because it's only fed to the queen. And that's what makes the difference between a worker bee and the queen. They're both females. The males do not do any work. So an egg that has been laid within the last 3 days that is fed royal jelly will develop into a queen rather than into a worker bee. And here's some royal jelly that's been harvested along with the propolis, along with the pollen, and mixed together, and these are supposed to provide many health benefits. Well, here you see the result of an egg, fertilized egg, being fed the royal jelly. So letter B refers to the queen cells of developing queens that were fed the royal jelly. C are the drones, the males, the drone cells. They're larger than the regular size cell. Those are unfertilized eggs. We'll get to that. Letter A is a developing worker bee, a fertilized egg, but not fed the royal jelly. And there at letter D, you can see an egg that's just been laid within the last day or two and still has the potential to become a queen if the workers decide to feed it the royal jelly. So the workers actually decide if an egg is going to become a worker or a queen bee, depending upon the needs of the hive. Well, when the bees have been doing very well, they've had great sources of nectar and pollen, they're able to multiply like mad and become crowded, then what they'll do is they'll take off with the queen, the existing queen, and fly off to form a new hive and leave eggs behind that have been fed royal jelly, so a new queen will emerge and be the queen of the original hive. And so the queen will fly off, the existing queen will fly off, land on a temporary spot. The workers will know where she is by pheromones, molecules that she emits. And then scouts, female scouts, will fly off to find the permanent residence. Hopefully not a hole inside the wall of your house. So back in the hive, back at the ranch, where these fertilized eggs that have been fed royal jelly have been developing in those queen cells, there are several of them because they don't just rely on just one happening. So the very first one that emerges will go find the other queen cells and will rip into them and then sting the other queens who are almost ready to emerge, so that she will be the only queen bee. And that's where this phrase queen bee comes from, because she will kill the other queen bees. Now, the queen does not have a barb on the stinger, so she will not die by stinging another bee, because she won't have her stinger ripped out, as opposed to the workers who do have a barb. And so here you see what's happened, where this bee stung this person, the barb caught in the skin, and the stinger, as well as some musculature and a gland containing the venom, remain in the person who got stung, or the animal that got stung. And those muscles will continue to pump the venom into the person, unless you have the smarts to take something like a credit card and scrape sideways to remove the stinger. If you try and use your fingers and pick up the stinger, what you'll end up doing is squeezing the poison sac and pushing all the venom into yourself. So whenever you're stung by a bee, you want to scrape the stinger away. So the loss of this tissue causes the bee to die. So she gives up her life to sting. So they don't sting unless they have to, unless they feel really provoked. As in this cartoon, why are you here in bee heaven? I was provoked and stung something. Hey, me too. And then this one bee says to her co-worker, he wasn't worth dying over, so I didn't sting him. I just headbutted his eyeball instead. All right. Now here you see in flight a queen being mated by a drone, and that's the only task that the males have, is to mate with the queen. They do no work, they gather no nectar, no pollen. They're fed by the workers. So they're maintained for the sole purpose of mating with the queen and providing the sperm to fill her sperm sac. And so when the male separates from the queen, his guts get pulled out and he dies in a blaze of glory. And that's the end of him. So the males have the choice of dying by mating or dying by starvation at the end of the season in the fall, when the nectar is not going to be available anymore and the workers kick the males out of the hive. So here is the fertilized queen, and she mates with multiple males in flight, and this is at very high altitudes as well. Kind of a test as to which males have the strength and the ability to get up that high. So the queen then inspects each individual cell to make sure the workers have cleaned it out properly from prior use. Then she will lay an egg into the cell. If the cell is a worker size cell, the most common size, she will fertilize the egg. So a fertilized egg becomes a female. Excuse me. If the workers have provided cells that are larger, the size for drones, she will not fertilize the egg as it leaves her egg sac and will become a male. So the workers decide who's going to be what. They decide if it's going to be a male or a female or a queen, and she just simply lays the egg accordingly, whether to fertilize or not to fertilize. So here you see the newly laid eggs in the bottom of the cells, and you can see the discoloration from the propolis, a thin layer of propolis that's been put there. And here you see the drone cells. Those cells are much longer than the normal cells, so they are protruding in this spherical fashion. So there's 3 drone cells there on the left. And you see how much larger the queen is than the workers, and you see how much larger the queen cells are there as well. Takes 16 days for a queen to develop, and that's a much shorter time than for the males or the workers because of that royal jelly, providing a much richer food source. 21 days for the males. I'm sorry, for the females, and 24 days for the males. So there's a significant difference there in the time it takes for them to grow to maturity, to emerge. And here it's showing you the difference in the chromosomes. As I said, the unfertilized egg for the male, so it only has one set of chromosomes from the egg, whereas the females have a double set, one set from the egg and one from the sperm that's been released. So here at the end of the pollen and nectar season, the drones are kicked out and they starve to death out in that cold, cruel world. What a terrible way to go. Well, here in scripture, there is a reference to a person who is a drone, idle, disruptive, not busy, busy bodies. Okay, such people, we command and urge you to settle down and earn the food you eat. In other words, no worky, no ticky, right? Your Majesty, the workers are discussing the possibility of forming a trade union. Uh-oh, rebellion. Well, the word honey appears in scripture 61 times. Reference to the land flowing with milk and honey 21 times. And there's a riddle from scripture. So this is in Judges. And so Samson is saying to the men gathered, out of the eater came something to eat, and out of the strong came something sweet. So he made a bet with them, and he, made a significant bet, and that they would not be able to solve the riddle. So for 3 days, they, trying to figure this out. Then on the seventh day, they went to him and said, what is sweeter than honey, and what is stronger than a lion? And he said to them, if you had not plowed with my heifer, meaning Delilah, you would not have solved my riddle. So she had begged him, tell me the riddle solution. And you don't love me if you don't tell me this, stuff. So he told her, and then she spilled the beans, and so they won the bet. He got mad. Here's a picture, a representation of a lion's skull with the honeycomb inside. And you know what happens after that. The temple goes down and all sorts of stuff happens. Well, the properties of honey in terms of medicinal action, there's the antibacterial actions we mentioned with the hydrogen peroxide, the acidity, making it so that the microorganisms are less likely to be able to grow. Hyperviscosity means super thickness. It's still a liquid, a fluid, but it's very, very thick. And there's the phrase, someone who is slower than molasses in January. Well, molasses is a sugar solution from sugarcane, and it's very thick. It's been concentrated. So same idea here. It forms a physical barrier just because it is so doggone thick. So you can put it on a wound to help provide a physical barrier to protect it against infection. In addition to, because it is such a concentrated solution of sugar, it has what's called osmolarity. In other words, it's going to draw water to it from a place that has more water. Well, this would be in the instance of putting it over a wound, an open wound, drawing the fluid from the area where the infection has occurred already, and taking that fluid, that inflammation, that swelling out of the area where the inflammation is going on. I already mentioned it's the physical barrier, if it's applied immediately, keeping bacteria out. So here you see this representation of the wound with the inflammation with the pink color, and the fluid, the inflammatory fluid going into the wound because of the normal processes, the body with inflammation. And then this type of honey called manuka, it's from a particular plant called manuka plant, forming the physical barrier to keep anything else from getting into the wound, and drawing the fluid due to the osmotic action out of the infection, helping to deal with the bacteria already there from the original wound. So all of these things, and then it also stimulates cells in our immune system called monocytes. So here there's a diagram of the tissue, and you're going to see a thorn enter from the top right corner in a second here. This is showing you what a monocyte looks like in the bloodstream. It has a very particular shape to the nucleus. It's very identifiable from the other kinds of white blood cells. And there you see the thorn entering in. I'll have it happen again. Here's the thorn coming in, introducing bacteria into the tissue. Now there's the inflammatory process represented by that pink discoloration. And then now these monocytes are stimulated by the inflammatory process, but even more greatly by the Manuka honey, to leave the circulatory system and become what we call macrophages. They change into macrophages, and the word macrophage literally means big eater. So these macrophages migrate through the tissue between the cells to go where the inflammation is occurring. And here you see this macrophage, very large cell, here engulfing, surrounding these bacteria. You see these kind of rod-shaped bacteria here in chains being gobbled up by this macrophage. It's part of our immune system's job. And so the Manuka honey increases this process, drawing the macrophages here. Well, these macrophages also secrete a molecule called tumor necrosis factor alpha, which also is part of this inflammatory process. And this thing will help kill the bacteria. This molecule will punch cells in the walls of the bacteria and help the bacteria to die. In addition, there's another molecule called B-defensin 1, which is present, and this helps speed up the healing of the tissues by increasing the migration of the cells that make up our superficial layers of our skin to speed up healing. So it has good action against the bacteria and good action helping the body to actually restore the normal cells there that have been destroyed by the infection. Well, here's just an example of some of the types of infections that are bad news that do respond to these antibacterial properties in honey. And these are serious and even life-threatening infections. Well, there's another plant here called ulmo plant that also has microbacterial activity, and against one that you hear a lot about called MRSA, methicillin-resistant Staphylococcus aureus. That's the big long name referring to this particular type of bacteria, which is highly resistant to antibiotics that we use and can be fatal because of that. Well, this is the plant, the ulmo plant, from which the honey is derived, that is effective against these resistant bacteria. And this here you see here is the place in very southern part of Chile there where this plant is found. And so they grow this plant to harvest, to have the bees harvest the nectar to make the honey, which is then sold for medicinal purposes. And it has especially high concentration of the hydrogen peroxide, which is a major reason why this is more effective in treating these infections. So here you see an example of this infection on this foot here. And then the honey is applied to this and this person recovers. So nowadays, here even in the US, honey is being used in wounds. Now, this is a relatively new thing in part of mainstream medicine. This was not done years ago when I was going through training. So this is a new tool that we have to fight these infections. And here you see further things listed that can be dealt with as well. Well, here is the manuka plant for the manuka honey, and beautiful flowers. And this is from New Zealand. So you see these little dots indicating where this plant grows in New Zealand. And these are the types of molecules that are isolated from this honey that are effective in fighting infection. Well, in particular, this one type of organism is called Helicobacter pylori, and this is known to cause stomach ulcers, gastric ulcers, duodenal ulcers, the first part of the small intestine immediately after the stomach. And years ago, it was not known that these bacteria caused this ulcer. It was just thought it was the issue of stomach acid all on its own. And in those days, patients were told to drink a lot of milkshakes to coat the stomach to help protect it against the acid in the stomach. But it turns out that was the worst advice possible, because of the calcium in the milk, and the milkshakes actually further stimulated acid secretion and exacerbated the problem. But that wasn't known at that time. Then an Australian physician doing research figured this out, and nobody would believe him. All the medical community said, "Oh, you're ridiculous. You know, this is, how can this bacteria cause an ulcer?" Well, he was so sure of his work that after he had proven to his satisfaction that he had the appropriate antibiotic to kill these bacteria, he then drank a solution of the bacteria itself so he could develop the ulcer, document it, take the antibiotic, and document the healing with the antibiotic. And then physicians became believers and said, "Okay, you've proven this, you've done the scientific method on this, and all right, we were wrong." And so now antibiotics against Helicobacter pylori become standard part of therapy. Well, manuka honey is one of these things that, when ingested, is very effective in treating this bacteria. Now, this shows you what happens when the bacteria erodes the tissue to the point where it compromises a small artery in the lining of the stomach. And so you see here, under arterial pressure, the blood being pumped out of the artery into the gut itself. And then this is why people with these kinds of ulcers end up with black stools, because of the change of the color of the blood as it goes through the GI tract. So this honey can be used to treat these things, as well as antibiotics that we use as well. Well, it's found that honey actually does a better job against colds, the common cold, than anything else that we have. So the recommendation to use honey for the common cold is actually beneficial. And so here's just a quote from the article showing about how this works with the viruses that cause the common cold. So a question to ask yourself is, how is it that this multitude of enzymes, antibacterial, antifungal, antiviral agents came to be in honey by way of the secretions of these several glands in the bodies of the worker honeybees? So I didn't quite explicitly say that when the bees regurgitate the nectar into the cells, that's when the secretions from these glands enter into that nectar as well, and becomes part of the honey as the nectar is evaporated, concentrated through evaporation. So did all of this stuff happen by a series of random chance events, or is it by careful design by our omniscient, all-powerful, omnipotent creator of the universe? You know, which is the most likely probable situation here? So in summary, amber has worldwide distribution. There's a fascinating history in northeastern Europe. The ecological zones of the entrapped organisms, marine, freshwater, as well as terrestrial. The mechanism of formation needing to be in a water environment so things can be entrapped and entombed, totally encased. Evolutionary difficulty in assigning dates. The dating headaches caused by bees with the flowering plants 100 million years. And then about the bees, how they detect aromas, how they see, how they communicate, transport nectar and pollen. We can even see how they stimulate the plants to release nectar and pollen, how they process the honey, how they reproduce individuals and colonies, and then the antibacterial and wound healing properties of honey and also of pollen. So we've covered a lot of material. And so the one bee says in the original Gary Larson cartoon, "Face it, Fred, we're lost." But what's the problem with this cartoon? The workers are females. So it should be, "Face it, Frederica, we're lost," as they look at the map of the honeycomb. All right, so this is the time for questions. And I don't know, Jim, if you're going to be able to get people's sound turned on so I can hear the questions or not.
Let me see what I can do.
I'll look and see if there's some way that I'm blocking it, but I don't think I am.
You don't hear me, right?
Let me see, what else can I do here?
Get back on the phone with Jim and he can tell you.
Can you guys hear my voice?
Tell you what, I'll call you, and then if you can hear their questions, you can relay them to me.
Okay.
Patty's excellent suggestion.
Okay.
Okay, are you there?
Yes, can you hear me?
You're muffled, but yes. Yeah. Okay, so hold the phone down here.
Is this better?
Okay, I can hear you now.
Okay. Bob Schober, can you unmute yourself? And Tom Canfield, can you unmute yourself?
I think I'm unmuted. I should be unmuted. I don't have any questions though. It just is amazing.
Yes. And Tom Canfield, can you unmute?
What was that?
Tom Canfield, can you unmute? Yes, we have, well, hang on. Oh, all right. So my screen shows myself to be unmuted, Bob R to be unmuted, and Bob Schober to be unmuted. Tom Canfield still is muted. Okay. If anybody has a question, ask me. I can hear you, and I will relay it to Joseph.
I have a question.
Okay.
Well, it's actually more of a comment. Would it be a good idea then when I'm stealing the honey from the beehive and I get stung to then apply the honey to my wounds?
Okay. Bob Reinhardt wants to know if he gets stung robbing a hive of its honey, can he apply the honey to the wounds and will it help? Okay. Yeah. Wow. Well, you're lucky to be alive, you know. Yeah, but bee toxin is cumulative. When I worked at the Moody Institute of Science, I was there after they had finished their bee movie that they'd worked on for 10 years. And my immediate boss, um, they had actually made the movie at a different location before they moved to where I worked with them. And they had kept their glass hive up on the roof. And so over the period of 10 years that they worked on this, my immediate boss got stung enough times that he developed an allergy. And the last time he got stung, he barely made it down the stairs. By the time he got to the bottom of the stairs, both eyes were swollen shut and he was having trouble breathing. And they rushed him to the hospital and he almost died. But, uh-huh. Well, okay. Ah, okay. Okay. Well, let me give these guys your answer. Um, uh, he says that because bee stings are puncture wounds, they very rarely get infected. He's been stung hundreds of times and has never had an infected bee sting. Um, the the benefit of the honey would really be if you did have an infection and the honey could get in to clear up the infection. And then in response to what I just told him about my former boss at Moody Institute of Science, he said you can develop an allergy after one bee sting or two bee stings. It isn't necessarily after many, many bee stings. So it's just a question of when you get your allergy, when they become dangerous. You know what? I'm going to put you on speaker, and presumably they'll be able to hear you over my phone. Okay. Okay, say something and then you guys respond if you can hear him.
I can't make out what's being said.
Okay. Were you guys able to hear Joseph when he spoke to me just now over my phone?
I could hear it.
Okay, Bob Schober heard you. Bob Reinhardt? Oh, he's gone.
Says Joseph is on mute.
Yeah, he muted himself to avoid feedback.
Right.
Okay, yes, Joseph. The only one with his mic still open is Bob Schober, and he hears you fine.
Okay, so any other questions?
I jotted down questions as I was listening, but you answered them in the course of the rest of the presentation, so I don't guess I have any.
All right.
That was absolutely fascinating. Lots and lots of good information I'd never heard before.
Well, I'm glad you enjoyed it.
Certainly did.
All righty, so I guess we're kaput.
Okay, we'll close it and I will stop recording and put it up on the website.
All right. I don't know how much of this tail end stuff you want to include since it's so technologically goofy. All right. Well, all right. Thank you very much for having me, and we'll see what happens
All right.
Thank you too. It's
Yeah.
just this great stuff.
All right.
Okay.
Lord bless.
Okay. Looks like it.
Okay, stop the recording. So Bob, how you doing?
Okay. Just pretty with the horses. Gotten fairly busy with some stuff. It's the technology I'm doing. What we did is we kind of looked at the way the neural system works, and we're trying to make electronic models of it.
Yeah.
And eventually I'd like to talk to somebody that knows something about it. And yeah. Okay.
And Tom, how are you doing? Good.
Okay. Normally, electronics take voltage in, and what I've done is I'm taking charge in, so I'm sensitive to a charge transfer. So looking at neural receptors that get a certain amount of charge and they trigger, so I can do that, and it's got really high sensitivity, high linearity, high speed. It's real compact, and been doing some fairly neat things with it. No.
But the idea is it appears to mimic neural transmissions.
Right. In other words, normally you're looking at voltage on a circuit, but neural looks at the charge transfer. So this is a charge transfer operating kind of electronics.
So you need a friendly neurologist to talk with, right?
Yeah.
I don't think. I don't know. I can maybe put the word out and see if we have anybody. I don't think there's anybody in Logos that specializes in neurology, but they might know somebody that does.
Yeah. Well, we got a book that's I don't know, it's like 1,500 pages. It's really kind of neat. We've been looking at it.
Dad, I'm trying to get you to sleep.
Yeah. No. I'll have to get with you and talk to you more about it. Okay. Guess that's about all I have.
All righty. Okay. Well, Lord bless you guys.
Okay, and, Lord bless.
Hopefully next time we won't have these glitches. That was crazy.
Yeah.
Hey, Jim.
Yeah.
Now that Calvary is allowing getting like we did pre-COVID,
you're breaking up real bad. Can you see it again?
I'll repeat that. So now that Calvary Chapel is doing in-person meetings again, any thoughts on us having our normal meetings where we meet in person?
Well, I will ask. If we can do it, we'll sure do it.
Well, I went to church last Sunday, and they said they're resuming all their
Oh,
yeah,
okay.
last Sunday they were resuming all their regular meetings this week.
Okay, I didn't
Anything
know that.
under like 100 people is fine, they said.
Good.
Yeah, so you may want to talk to the office, but it sounded like the regular meetings, like the women's groups and so forth, they're all going to start meeting again
Yeah.
in person.
Great. I didn't know that.
That would be great.
Very good.
I haven't seen you guys in so long. It would be great to meet in person again.
I know.
Yes. Starting to forget what you look like.
Yeah, I was just going to say that.
All right, real good. Well, let's close it up. Lord bless you guys.
Okay.
Thank you. Have a good night.
Okay.
Okay, good night.
Good night.